Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 148th seminar, and I call it on ETH Zurich Day. I have been at my St. Louis home for 2 weeks without traveling, which allowed me to spend time with my family for the Thanksgiving week and this week. It is so good to have such family time with all three of us, including my daughter, given my crazy trip this year, which end up more than 200 days out of town with a majority out of the country. I will embark on the last business trip to Hawaii tomorrow to host the first international SynBYSS conference, followed by some potential winter break trips with my family again. Looking forward to seeing many of you in a paradise island to discuss the synthetic biology future and then finally get together rather than only two people talking each other weekly. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Sai Reddy. He is a professor at ETH Zurich. I believe I met him for the first time during my postdoc interview in the Georgiou lab at UT Austin. I received the offer, meaning that I might have become his academic brother, but I chose Chris Foy in San Francisco at the time, considering my wife's preference regarding the location, probably warmer, not warmer, actually the cooler, uh, temperature city than Austin, I believe. I met Sai again last year at a conference, and I invited him to give a talk after I listened to his amazing talk, also amazing science. Sai, thanks so much for accepting my invitation and your great contribution to the scientific community with amazing scientific achievement and then contribution again. And the virtual podium is all yours now. And thank you again.
Thank you, Tae-suk. It's a pleasure to attend this and present, and looking very forward to Liz's presentation as well. I will start to share my screen now. Okay. So today I'm going to give, of course, a very short presentation with very little data, but I really want to talk about the idea of drug discovery and how this is really important to develop a process for immune cell therapies. Okay. So I'm going to start by just introducing how typically the drug discovery and clinical development pipeline looks like. And this is a pretty generic figure, but if you did a Google search, you'd probably find many like this. And the idea in drug discovery, typically, whether it's small molecule or biologics, you do a sort of screening experiment. There are different kinds of screens, obviously. In antibody or protein biologics, these are screens could be things like phage display, yeast display. Small molecules, there are different types of arrayed screens. And eventually you do some large scale screening to find candidates. And then you move on to a subset of these candidates that then go through some very, detailed characterization, eventually relying, usually in the industry, this is a multi-year process that gets you down to candidates that you move through the clinical development pipeline. Eventually, if successful, you get approval. And why I bring this up is I'm going to come back to this. It's actually going to be my last slide, but I just want you to think about this for a second. Keep this in mind as I now transition to really talking to you about cell therapies. So T cell immunotherapies are really one of the most promising areas in this field. And the idea is simply that T cells play a particularly important role. There are many different kinds of immune cells that can interact, but in particular, T cells like CD8 T cells can actually interact directly with things like tumor cells and lead to clearance. There are other types of cells, NK cells, macrophages, and dendritic cells that can also potentially be used for immunotherapies, although those areas are not as well developed. So if we focus in on the T cell, one of the ways in which it can interact with the tumor is through its T cell receptor, which can interact directly with a peptide HLA complex on a tumor cell. This could be through something called the tumor-associated antigen. This could drive T cell activation, which would result in a cytotoxic response that eventually leads to tumor cell killing. So this is, and in the field of immunotherapies, an idea is trying to enhance and redirect T cells so that they have a specificity for cancer cells in particular. Okay, so move on to the next slide. Now, in this field, where the most advances have developed is something called chimeric antigen receptors. And this is a very exciting area for synthetic immunology. This consists of the idea of these are synthetic receptors in which the antigen binding fragment of a monoclonal antibody, it's highlighted here in red, can be selected, and it can be fused with the intracellular signaling domains of the T cell receptor complex. This ends up in this essential, as well as other co-stimulatory domains, which can be involved in T cell activation. So these are all linked together in this synthetic receptor. So this red part is an antibody, basically, which can, of course, bind to an antigen. And these are intracellular signaling domains from T cell receptor signals and co-receptors. And so this allows one to repurpose a T cell to recognize a tumor cell, as shown here. And then this would lead to essentially a cytotoxic response. Now, this is probably one of the most successful examples clinically of using this approach of synthetic receptor engineering for immunotherapies. And this field has been around for a long time. It's been developing in many ways, but it's only been in the last, let's say, 5 to 7 years in which we've seen quite a bit of success. There are now 7 FDA-approved CAR T cell therapies. These are all essentially used for blood cancers, typically hematological malignancies like lymphomas and leukemias. And they've led to tremendous success, in some cases leading to patient remission and essentially a cure for patients. And these are typically used on patients who, as a last line of treatment, who are essentially unable to respond to any other types of therapy. So it's been a major success. However, it's not without its challenges, as anything might be in drug development. And there are multiple challenges in this field. One of them is that not all patients actually respond. So there are a subset of patients who get this treatment and who don't actually respond carefully. Another challenge is toxicities. The concept is basically engineering these cells ex vivo and then reintroducing them into a cancer patient. This is a massive, massive introduction. Billions of cells typically are transferred into the patient, and they lead to this huge inflammatory response because they immediately start killing tumor cells. And this can lead to a pathology called cytokine release syndrome, as well as neurotoxicity. Patients have unfortunately died in some cases or undergone severe side effects. So toxicity is still an issue. And probably most notably, one of the unmet medical need is the fact that all CAR T cells today that are clinically approved are against liquid cancers. So basically blood cancers. There has been a lot of effort in recent years to move this into solid tumors and other types of cancer, but with limited clinical success to date. And it's questionable whether the current strategies of CAR T cell therapies will really be successful against solid tumors. And so that's probably one of the biggest areas in which this field needs further innovation. Okay. Sorry. So one of the things I want to focus in on is the CAR signaling architecture and how this might impact the functional properties. Now, the signaling architecture, if we take a closer look here, As I mentioned, there's a signaling domain. And one of the primary signaling domains that's always used is called the CD3 zeta domain. This is essentially linked to the T cell receptor signaling complex. So that's always used. And then another domain that's often introduced is either CD28 or 41BB. These are co-receptors of T cells. So these signaling domains, they offer, in some cases, more potent effector functions, including, differentiation and cytokine secretion. That's CD28. 41BB tends to be associated more with T cell persistence and a slower, more gradual response. So this choice and number of signaling elements actually can make a big impact. However, when we look at clinical stage studies, you can see that basically the vast majority of these therapies have been only using 41BB and CD28 or both. And only about 6% have ever explored alternative signaling domains. And so the question is whether when we think further and we really look at T cells, they're an incredibly complex signaling network exists. There are many co-receptors and cytokine receptors and chemokine receptors as the like. And these all have, of course, they have extracellular domains, but signaling domains as well. And so this is an extremely complex network. And you can kind of look here as a very nice example of some of these different properties that might be associated with some of these receptors. So really, one of the questions we have is, can we expand on these cytoplasmic intracellular signaling domains to make completely new types of CAR T cells that have different type of phenotypes, which might eventually lead to solutions for those existing problems, like solid tumors, like less toxicity, and further on. So when we look at how traditional CAR engineering is performed, it's essentially done very rationally. So one would look at, what are these domains that are involved, CD28, 41BB, and they're put together and they're tested one at a time. So that's, for a large extent, that's how drug discovery for cell therapies is done. It's done by a very rational, one at a time approach. And of course, this then requires extensive preclinical development. However, the thing that I'd like to really emphasize in the last couple minutes here is that we are emerging on an opportunity to start doing high throughput CAR engineering. And the idea of this is to actually take libraries of signaling domains, modified or shuffled in different ways, and introduce them into T cells, and then do pooled screening assays and then measure in many more elaborate ways what is exactly happening to discover new combinations that provide new opportunities for phenotypes that have desired properties. And while our group has started to work in this area, quite a few groups have. And I want to point you to some very exciting publications from colleagues in this field. I could just start out here. This is a study from Michael Birnbaum's group at MIT. They did a very impressive work where they actually took 700,000 signaling domain combinations and screened them in a JERCAT T cell line, and then eventually did different approaches for screening. Cole Roybal's lab did a different approach where they directly went to primary T cells. Wendell Lim's group did a very exciting study where they used different motifs of signaling domains and also applied kind of a machine learning framework to screen a larger number of variants in silico. So, and then I want to highlight our two studies that we've put out. And in our two studies for my group, we've done things related to where we do directly primary T cells. Interestingly, we're the only ones so far where we put it directly in with CRISPR-Cas9 CAR T cells. And then what I really want to highlight is our screening is based on single cell RNA sequencing. And I'll get to that in just two more slides now. I'll explain it on this. And I'll highlight this study. We have another one that should come out online, but is also on bioRxiv. So our workflow, as just an idea of how one can do high throughput CAR engineering, is we did a big library of signaling domains that are assembled, cloned together, and then introduced by CRISPR-Cas9 into primary T cells. These pooled libraries of CAR variants are then co-cultured directly with tumor cells, expressing the antigen. And then instead of like doing a functional readout, like, there's no fluorescent reporter we're looking for, we're not looking for a phenotype. We actually just take the T cells after exposure to cancer cells and just do single cell RNA sequencing. And then what that results in is this large transcriptional profile that allows us then to profile this large library based on the T cell phenotype. When you think about it, T cells are, if they are the medicine, then their entire transcriptome and profile is part of the medicine, and that's far more interesting to look at than just a single parameter, like do they kill the tumor cells? So this allows us to get this very large scale introduction. And eventually we can identify profiles, even map them onto like clinical data to maybe identify combinations and new variants. And just to show you how we did this, we took a set of signaling domains here and then essentially shuffled them with other signaling domains all together. This allowed us to create, this is all done by Golden Gate cloning, to eventually have 180 variants as a pooled library, which we then screened, as I mentioned, by doing co-culture with tumor cells and single cell sequencing. And this is the output of the single cell sequencing data. This is just a UMAP of the transcriptional profiles. And you can see we have different areas of CAR T variants that have different profiles. These are T cell phenotypes. And one of the most interesting might be activated and cytotoxic. So these can be then, we can look at different variants. So these letters here, A, B, C, D, et cetera, are different variants and different profiles. And then 28 and 41BB here, these are what we might call the control CARs, have this type of profile. So we can even see ones that might have more of a cytotoxic profile than the ones that are used clinically. And then what's interesting is even though all of this was just a single cell RNA sequencing analysis, we then selected these variants and then did then in primary T cells and then did with individual variants tumor cell killing assays. And what you can see here in blue are the clinically used CAR architectures. But then we found new variants that in some cases lead to faster tumor cell killing, as well as we did some cytokine profiling as well. So really it shows you that there's an opportunity to bring a screening process into this whole field. And really this is basically what my vision is to say that in the future, immune cell therapy should go through a drug discovery process that looks similar in some ways to the other areas of drug discovery. And instead of screening just a few variants by rational design, ending up with these large libraries and very unique and exciting ways to do screening, maybe using single cell sequencing and other assays might lead us to a much more productive field that has unique variants that really solve some of these problems, the same way that drugs in many cases have solved problems not through rational design, but through the screening process. And so I'm excited that hopefully in the future we move in this direction. And then with one last slide, I want to of course, thank all of my great team that have worked on many projects. I want to acknowledge in particular Rocio and Rafael. Rocio is an outstanding PhD student that really led these projects on CAR T cells, as well as Rafael, who's now at Regeneron. And if I'm allowed to give Tae-Suk one advertisement, I also want to announce that recently we launched the Botnar Institute of Immune Engineering. So it's a brand new nonprofit research institute that's focused on the advanced study of immunological systems. We have integration of systems, computational, and synthetic immunology. We will be moving into a new building in Basel, Switzerland, with partnerships with ETH Zurich, as well as with the University of Oxford. The BIIE is also very fortunate to have received a very large donation of $1 billion US from the Foundation Botnar, which is a philanthropic organization who is focused on improving the health and well-being of young people worldwide. So the BIE will also have a specific emphasis of also advancing global health and child health indication. So thank you very much. And as we develop the BIE, be happy to engage with future many synthetic biologists.
Thank you. Absolutely amazing. So, one day, someday, very, in near future, I want your technology or many other technologies, lead to many, safer, cancer therapy method, because a lot of people suffer from, cancers, that their family also suffer from cancer, including myself. So I hope that end up, great solution near future. Thank you.
Thanks.
All right, so now the main speaker of today with a longer introduction. Dr. Elizabeth Wayne is a TED Fellow and currently an assistant professor at the University of Washington, Seattle. Prior to moving to Seattle, she was at Carnegie Mellon University in chemical engineering and biomedical engineering. She works at the interface of immunology and nanoparticle engineering by exploring the function of monocytes as a cellular theranostic. Her group designs chitosan-based nanoparticles that can modulate macrophage phenotype under inflammatory context. Her group designs monocyte biosensors to enable real-time measurement of macrophage polarization. Currently, she is applying these concepts to investigate air pollution, lung regeneration, cancer, etc. Her research has been recognized through funding from NSF, Genentech, and a prestigious NIH R35 Maximizing Investigators Research Award, also short MIRA. She is also a science communicator who uses her platform to discuss the future of immunoengineering and issues related to underrepresented minorities in science. In 2017, she gave a TED Talk on immunoengineering, which currently has been viewed over 1.5 million times. She was featured in the Super Cool Scientist, a woman in science coloring book. She's also the co-host of the show PhD Divas, a podcast that tells the stories of women in leadership and higher education. Dr. Wayne has been interviewed and written in various platforms, including PBS NewsHour Brief, but Spectacular Series, Aspen Ideas Health Festival, Nature Careers, Nature Medicine, Bust Magazine, The Atlantic, and the LA Times. She's a true rising star or science celebrity, I would say, and it was my pleasure to meet her last year at a conference and after her amazing talk. Liz, thanks so much for your time today. Please take it away. It's all yours now. Thank you.
Thank you. That was a wonderful introduction. I hope I live up to it. And yes, very excited for this platform and this opportunity. So I want to talk to you guys about why I have an unhealthy obsession with monocytes and macrophages and how I think that they can be used to cure all, let's say. So broadly in my lab, we're interested in how monocyte phenotype can drive drug responses and how we can use them for drug delivery and design. So we do experiments where we characterize monocyte and macrophage interactions. Again, to think about how we can tailor both responses to improve drug delivery. We develop platforms that allow us to do spatial temporal imaging of macrophage responses in real time for diagnostic purposes. And we also think about how we can use data generated from either of these processes to create a closed loop circle that can lead to better design. So I think a lot about nature and nurture right now. This has been my tissue engineering project, the best one I've done so far. And I think about a lot his future and about fate. Who is he going to be when he grows up? And I no longer get to put on these orange pumpkin shirts, sweatshirts for him anymore. And so I think about this a lot also because immune cells are very similar, where there are intrinsic and extrinsic factors that lead to how they perform in immune responses. And so there's one idea of nature where, he has my eyes. I think genetically I gave him certain things. And then there's also nurture or environment, where I cannot tell you how he decided to try to climb a Pampers box head first, but that is where we were. And that's where he likes to be. And so that is all him, but the environment is there. He decides to do those things with the environment. And so cells are also very similar, where there are some genetic, epigenetic, but intrinsic factors. And then there's extracellular factors like the environment around them, like matrix, um, cytokine signaling, that then shape them into the cells they become. And then those responses are what is the difference between acute inflammation, chronic inflammation, to fibrosis, you name it. And so this is why I'm interested in them. And so macrophages are also interesting because they are innate immune cells and they are found in every organ. And so these are cells that perform phagocytosis, migration, secretion, chemotaxis, and they help initiate and resolve immune responses in these tissues. In the brain, they're called microglia. In the alveoli, in the lung, they're called alveolar macrophages. Liver, they're Kupffer cells. And what's also interesting is that although monocytes are not necessarily macrophages, they're not tissue resident macrophages, but they are systemic in that they migrate from the bone marrow, circulate through the bloodstream, and during inflammation, they can migrate into tissues and differentiate into either of these phenotypes, and again, help perform these localized immune functions. And to kind of piggyback on what Dr. Reddy was talking about earlier, they're also interesting in the cancer context because many solid tumors have large masses of macrophages that also help control immune responses, in most cases for worse in terms of immunosuppression. But we can also think about how we can use those for drug delivery in tumors because they can be modulated to be pro or anti-inflammatory. And so how do they make those decisions? And one of the things that I mentioned were extracellular factors. And so here's an experiment, a time-lapse experiment, where I gave these cells either interferon gamma, which is a pro-inflammatory cytokine. And in these environments, you see that these macrophages are actually now phagocytosing these other potentially dead cells here. And if you look closely, if you know anything more about macrophage biology, you also see there's little granules in them. So they're becoming more phagocytic and also able to increase their lysosomal activity to degrade those. If I give them media from cancer-conditioned media, what you see is they then begin to form this kind of elongation, very much slower moving, and they are not phagocytosing as much. They're kind of becoming what I call couch potatoes, very immunosuppressive. But suffice to say that these cells are naive first. I gave them two different cytokines and I let them make these decisions. And so there are signals that go in and then something that happens out as a result. And as an engineer, we like to think about how we can actually design these signals that can help control or at least modulate or influence monocyte responses. From the drug delivery world, we think about nanoparticles, which are these small punctate objects that can encapsulate drug um, in various ways, either on the inside or they can be functionalized on the surface, but we can control basically everything about this nanomaterial. So we can control the size, the shape of them, what's inside of them, what are they composed of, whether that's a polymer or a lipid or some sort of silicon particle. And all of those things drive how they interact with cell surfaces. Now, cells are also very complex in that they have various receptors that actually help them bind or migrate or respond to any kind of immunological cues. And so it ends up being a bidirectional relationship where we can actually think about not only how do we design nanoparticles to tailor macrophage responses, but then what are those unique features about the macrophage, those intrinsic factors that actually determine which particles they are more susceptible to interact with. Okay, and let me just check this. Okay, sorry. Thanks. Okay, and so I want to actually show you a quick vignette of how I thought about how the polarization or how the current state of the cell influences their behavior. And this is a precursor to thinking about how we might design or better characterize these. And so here are nanoparticles in red. So these are siRNA lipid complexes that we can then visualize being uptaken by the cells. And what's also interesting here is that if you put these particles in cells, how they are activated beforehand really matters. So if you were to give them, similar to the video I showed you before, either nothing, which is the black bar, or we gave them interferon gamma with LPS, so things that make them very pro-inflammatory, things that make them think, oh, there's a virus or a bacteria nearby. They take up a lot of particles in comparison to these more anti-inflammatory functions of IL-4 activation, which is considered a wound healing phenotype, excuse me, or cancer condition media for more of immunosuppressed cancer phenotype. And you see they take up lower amounts of these particles. And if you look by intensity, you also see these M1 macrophages are phagocytosing quite a bit of these particles, which you might expect in a pro-inflammatory activation state. But if you do the reverse, and then you actually put particles into naive cells, and then you activate them with these cytokines, you also see they process these materials differently, such that the M1 and these M2 phenotypes have far less of these nanoparticles remaining. So the question is, where do they go? Did they get degraded? Did they get secreted? What is happening with these particles? So then we did more tracking. And here we're also looking at these different phenotypes and looking at either green, which is the marker for a lysosome, which is the garbage can of the cell. And we can see that when you induce them into that pro-inflammatory phenotype where they're uptaking and eating a lot of these nanoparticles, you see they're also degrading them. They're co-localizing nanoparticles with the lysosome. So they're being shuffled to the lysosome for degradation. But in these anti-inflammatory cases, they are not. They're actually not being degraded in these cells most frequently. And when we did further exosome studies, we also found that these M2 phenotype cells were actually more likely to secrete those secrete more exosomes in comparison to the M1 cells. And as a drug delivery person, we thought about whether this could actually be a way of controlling how cells can secrete drug. So we now, instead of using just a siRNA that had luciferase attached, using an siRNA that just was a reporter that had a dye attached, we actually gave them a luciferase, an siRNA that would knock down luciferase. And then we co-culture those with cancer cells that express luciferase, such that if there is actually transfer, so if in the anti-inflammatory phenotype, these cells are more likely to secrete exosomes, or are they also going to secrete in those exosomes, would they contain the siRNA that we loaded into them, suggesting that those nanoparticles are actually shuffling not to the lysosome, but to exosomal pathways. And we found that to be true, that if you look at the M2 group, they have far less of the siRNA compared to the M1, which had less transfer occurring. The control group are actually just the macrophages and cancer cells co-cultured to account for any cell death that might occur just by them being co-cultured together. And we looked at this in 2 different cell lines, and we found that the trend was still true, that the siRNA was being transferred, and even more so in this anti-inflammatory phenotype. We also then took the nanoparticle out of the equation altogether, and we thought, well, maybe this is something that cells are just doing. So with electroporation, you actually just use electrical pulses to create little holes in the cells that the siRNA can then go into. And then we found yet again that after electroporation and conditioning these cells, the anti-inflammatory groups expressed less of that siRNA. Less of the siRNA. And when we did the co-culture experiment with the cancer cells that express luciferase, you see that the anti-inflammatory conditions had far less siRNA remaining compared to this M1 phenotype. So we thought about how this can be used as one, as a drug delivery tool, because that was my initial interest. But what also became interesting to me was that the phenotype of that cell matters. And when you look at drug delivery, where we're trying to actually heal or regenerate or address a chronic inflammation problem like cancer, those cells are never naive, and it really matters what phenotype those cells are in. So this really led me to think about how do we characterize the heterogeneity of monocytes and macrophages? Because those phenotypes will actually tell us what they're doing with the drugs we do deliver. They will tell us, and they will actually give us information about how we might be able to design particles in themselves. So there are three central questions about heterogeneity of macrophages that I think are really interesting, and that is differentiation. So I mentioned that monocytes will migrate into tissues and then they can become either macrophages, and their litter's a little dicey on it, but dendritic cells or other types of myeloid depressor cells. And so how do cells differentiate and then perform immune processes there? How do they polarize, which is that they stay the same cell type, but they can modulate between pro-inflammatory, as in I am phagocytosing and actively fighting viruses and tumors, viruses and bacteria, in comparison to like an anti-inflammatory phenotype where maybe I'm here to wound heal, or I'm actually trying to suppress any type of inflammation or immune responses, both in the macrophages or in other cells. And there's this other idea of reprogrammability. And I think this is really important when you think about chronic inflammation. Are there actually arenas where cells have been so programmed into a certain phenotype? Can you actually reverse that phenotype and make them go into another cell? And how often can they do that over the lifespan of those cells? And so all of these questions that have real implications in terms of the perceived outcomes of therapeutic success are very spatial and temporal questions, which means these can change over time. They happen at fast or slow rates. And it matters whether they're near a blood vessel, whether they're in the heart of the tumor where it's most necrotic, whether they're on the periphery close to normal tissue or less stiff tissue. And we need tools to be able to measure that, again, to think about how we design these processes. So there are a lot of materials out here that measure cell state. And what I'm calling state is that you can measure, and today, at that very instant, you know what that cell is, you know the identity and how they're performing. And these are very powerful tools. RNA sequencing, whether it's bulk or single cell, proteomics, and these are techniques where you can assay the entire protein content of cells or the entire RNA transcriptome of cells. You could also do flow cytometry where you look at markers and then you say, of these markers, how many of them are expressed and how high? And also looking at histology. And these give you really nice snapshots of the cells. I think they're really great because they also provide large amounts of information. But in terms of addressing spatial, or not necessarily spatial because we have spatial transcriptomics, but the temporal aspects in particular, not all of them, but many of them require tissue homogenization, such that you get a pool of the responses rather than looking at single cells. You can lose that ability to do real-time measurement, which is to say that once you measure the assay of the cells, as transcriptome or proteome, you often have to lyse the cells, which means you can't see how they perform to another response. And these are all things that can limit the ability to look at the performance of cells, which is to say, how do they respond in environments and how long do they respond in that way? So at the time that we were doing this, we were really into this group, and we decided to name the technique that we were going for bioluminescent temporal spectroscopy, the ability to image over time to look at how cells differentiate and how they make those immune decisions in real time. And so BTS also stands for the technique of looking at these cells, but they also stand for really popular band, which I'm hoping that people know about. And this is a really great way to look at the spectrum of activity. So one of my really great students who now graduated is working in industry, actually designed these cells with transcription factor responsive element promoters. And the idea is that we want to know when cells are beginning to activate certain transcriptional pathways, how are they happening and when is it happening? So we have ones for STAT6, where when that pathway is activated, we see an increase in luciferase expression, similar for STAT2 and 1 and 2, and for NF-kappa B to look at various activity. And this becomes really important. So luciferase is also chosen because it looks really great. It's highly sensitive, and it also has a really short half-life, such that you can actually do some quantitative measurements. So I'll give you an example of why we think this is interesting. If you look at NF-kappa B, NF-kappa B is a master regulator of inflammation. It's activated often during pro-inflammatory responses. And we have here some pro-inflammatory cytokines that we know are activated by NF-kappa B, and no stimulation in the first column. And you can see if I do a one time point, you see maybe interferon gamma and this TNF alpha have similar responses or abilities to activate NF-kappa B, followed then by rizikimod, which is actually an immunomodulatory drug associated with the TLR7/8 pathway, and this LPS interferon gamma. But if you look at the dynamics of response, let's say if I take numerous time points of these, again, these same cells over time, you'll be able to see that the kind of curves or the shape profiles of their activation is different. And in fact, TNF alpha has a faster peak of activity compared to interferon gamma, which peaks at least twice half as long of time frame. And that some of these dynamics are actually really important in thinking about the kinds of responses that we have or what they mean in the body. The other interesting thing is that because you are measuring these over time, you can also calculate the baseline. And so you can answer questions about whether those cytokines were activated or whether this pathway was activated before the experiment started, because the initial a priori information about that cell is retained, and then you can measure these over time and do quantitative metrics. So what I want to show you is one experiment or two experiments that we did to look at this. And we were interested in one, in the question of differentiation. Cells, when a cell gets into a new tissue environment, how does it decide what to do? Well, it looks at matrix, it looks at cytokines, and there are three in particular that are really interesting, GM-CSF and MCSF. These help cells become monocytes to macrophages, and in specific types of either pro-inflammatory, anti-inflammatory macrophages. GM-CSF is more associated with inflammation or pro-inflammatory responses. MCSF is a proliferation survival signal, and you can see this being activated correspondingly. PMA is often used for differentiation in an in vivo context because it is a chemical modifier that's pretty consistent, often pretty cheap, so engineers like to use this. Activates NF-kappa B. So I looked at these three differentiation markers to see did anything about the rate at which to differentiate change. And what you can see here, if we look at CMV activity, which is often used as a marker for differentiation, or often used to track cell growth because it is thought to be constitutively active, that it actually during differentiation processes is not constitutively active. And in fact, depending on the cytokine used to stimulate differentiation, have different rates of growth. So in the gray is the PMA, blue GM-CSF, and red MCSF. And you can see that in PMA, they have a really slow descent. So they are downregulating their CMV activity and eventually being able to pick up their growth and have that characteristic kind of exponential growth curve you might expect. GM-CSF and MCSF both make that transition into differentiation to a normal growth curve at least 6 days earlier than PMA. So the way that they regulate these genes over time is different. Their growth rates are different. And what's also interesting is that if you were to then look at NF-kappa B and look at CMV activity in the same cells, you see they also begin to differentially regulate both of those factors, such that as the CMV is being downregulated, you also see this NF-kappa B being upregulated between 4 and 12 days after differentiation. And so PMA, we found, induces a faster peak in NF-kappa B activation in comparison to this GM-CSF activity, which actually does a later stage of activation, beginning around this 8 days and peaking at around 17, 18 days. Now, what's also interesting about this data is that these are also based on single doses of PMA or GM-CSF that were given at day 0, suggesting that when cells begin the differentiation process, it is a 2 or 3 week long process that happens, and that there are constantly genes that are being up or down regulated over time that then influence their fate. Okay. If you were to look at the NF-kappa B activation over time, you would get the recapitulation that this PMA activity in blue has a faster peak than the GM-CSF in green. And then MCSF does not really activate this NF-kappa B. In comparison, STAT6 is an anti-inflammatory phenotype, and you can see that over time, the PMA does not induce activation of STAT6 compared to the GM-CSF and MCSF. And this is also something you might expect. So if there's a cytokine that induces pro-inflammatory activation in these cells, they might then begin to preferentially activate pro-inflammatory transcription factors and then downregulate the anti-inflammatory transcriptional factors. Another interesting thing that can happen here is after you differentiate, if you give another dose of cytokine, in this case, these are STAT6 cells that are anti-inflammatory, and then if you give them IL-4 as a second signal, you then see these other activation curves where you can see these dose-dependent responses to IL-4, but interestingly, PMA had a very abrogated, less dynamic response to these cells, suggesting that the background at which these cells are differentiating controls their ability to respond to secondary responses in the environment. And so not only did PMA have a smaller response, so 10 ROU per protein compared to the highest of 50 in either of these cases of GM-CSF or MCSF. There was less dynamic range between the 1 and 100 nanograms per ml conditions, but also the length of time that they stayed activated was about 6 days compared to higher activation, even up to 12 days, where they actually remain activated compared to this PMA. So you got longer duration of response and then higher activity in different backgrounds. We also noticed that if you do cyclic activity, so instead of just giving a single dose, if you give multiple rounds of dosing, that you can actually look at how cells swing from pro-inflammatory to anti-inflammatory activation. So if TNF-alpha only doses, you see that they reach about 100 nanograms, 100 ROU. These are relative light units, so they're actual measures of that luciferase activity. And you see that they go down over time. So if you give TNF-alpha, they activate, and then they deactivate, and they activate, and then deactivate. Tissue conditioned media, there was little activation of NF-kappa B, particularly as you compare to the TNF-alpha, which is a known cytokine that activates NF-kappa B and macrophages and monocytes. And if you cycle them, you also see this damping activity. And so what's interesting here is that one, cells don't just usually pump and pump continuously. So they can become exhausted, they can become tolerant to signals, and you can see this in terms of them not actually reaching the same levels of their highest activation from that first probing. What was also interesting is that if you gave the cells tissue conditioned media and then TNF-alpha, they reached twice the amount of response, so this 200,000 ROUs compared to TNF-alpha alone when they had no activation prior, suggesting that the activation of the cells before helped them shift into higher activation levels. So again, these are all plated at the same time, so they have the same number of additional cells, and you're able to kind of look at these dynamics. And I think there's a really interesting context in the context of cancer where cells are often pro-inflammatory, and we are trying to design therapeutics that make them pro-inflammatory. So how often can you probe them? How long can they stay activated? And what dynamics control those responses? The final thing that I will show you is looking at how we might use this as a diagnostic tool. So what we did is we thought about how the cancer environment is very immunosuppressive, and macrophages can make up a substantial portion of those tumor spheroids. Then can we use these as sensors to look at how monocyte activation in those tumor spheroids change over time? And if we can use the quantification around those activities, such as I showed you before, to actually say whether the degree of immunosuppression that a tumor spheroid has. So a way to characterize these phenotypes. So we looked in 2 case studies of HCT116, these are colorectal cancer cell lines, MDA, MB231 cells, or their breast cancer cells, and we wanted to see if we can actually tell differences in the profile of their activation over time to have another way of assessing the immunosuppression of the activity. So when we co-cultured those cancer cells, and we looked at different ratios, so 10:1 is a high cancer cell ratio, 1:10 is a high macrophage ratio. We found that over the course of this experiment, which is very short, but we saw fast activation of NF-kappa B that then rapidly downregulated. And this activity, as you increase or decrease the amount of cancer cells, these cells responded less. So the other way to also think about this is that the cancer cells are inducing NF-kappa B activation in the macrophages. And that is why they are pulsing and having these responses. As you increase the amount of macrophages, there's basically less other cells that they can respond to. So there's less of that immunosuppressive signal to respond to. The bottom graph here are actually the fluorescence. So these are tracking the cancer, the macrophage growth here. So what you can see is that at the highest times of activation, there's fewer macrophages here. And then the signal is starting to decrease as the macrophage signal increases. Now, in comparison, if you look at the MDA breast cancer spheroids, you see that at the high cancer cell ratio, you don't see as high of a peak. And so if the other one reached 3 at their highest activation, these are around 1. And then suggesting that the cancer cells are not inducing as much NF-kappa B activation in these macrophages compared to the that more immunosuppressive activity. And in fact, you also see there's a higher creep at the end. So there's slowly activation that is happening. Now, this was also interesting because if what we know about NF-kappa B activation is that it can be fast and rapid, or it can be slow and gradual, and that these are usually associated with canonical and non-canonical NF-kappa B activation. So this work is still really early, but we're really interested in thinking about how we can look at these profile responses and looking at how we can assess tumor immunosuppression, looking at different patient phenotypes by using these immune cells as these perturbation sensors, and also following up with allowing these to be drug screening platforms where we can then add drugs to see how do the drugs change the immunosuppressive environment of the cells of the tumors, and how do we reverse immune environments such that we can either make T cells more permissive or design more factors into these macrophages to actually lead to tumor changes in tumor growth. We also found that if you look at the time it takes for them to have their maximum response, that this is quantified here, that the HCT116 had a faster reaction time compared to these MDA231 cells, and that it also didn't matter what the background was as much, although PMA had shorter, faster times to get that activation, which is similar to the results we showed in the just 2D culture experiments. So these are some of the metrics that we can design. And I actually think I'm going to talk about this briefly, but these are my last slides. And so if we think about the background and how these cells differentiate, we want to know, does the differentiation phenotype of those cells, so if they have been primed with PMA, GM-CSF, or M-CSF, do they change how they respond to the to the tumor cells when they're in co-culture in these organoid phenotypes? And what we see Again, HET116 are more immunosuppressive, and we found that they had a higher maximum response and an AOC, which is area under the curve, which takes into the totality of the entire activation response. In these high cancer cell environments, the PMA cells were the ones that had the higher response in comparison to GM-CSF and MCSF inducing higher responses in the macrophages, and in the PMA not really inducing responses. So this also corresponds with there being different responses in these cells. If we then look at the opposite regime where we had high macrophage activity, then you see that these backgrounds weren't as specific. The PMA still had higher activation, but there weren't as higher activation in the HET116 cells, also suggesting that there was just less activation because there were fewer cancer cells around. So this is like a venue of the work that we've been doing. We're really interested in continuing both the development of these macrophages as sensing platforms, looking into how we can apply more systems immunology approaches, computational approaches to look at how we analyze these curves, and interested in how does the polarization in real time affect these responses that can lead to better design of drugs. I'd like to thank my lab and the current funding that we have. And if you have questions, I'd love to take them now, but also looking into the future. Thank you.
Fantastic talk. Thank you. Let me ask question first while waiting question from audience. So this is absolutely wonderful, important work. As I mentioned earlier, a lot of people suffering from tumors, cancers, whatever. And I see, two talk regarding cancer. Oh, by the way, I love your BTS slide and your slide. That's a wonderful. By the way, I'm MTS. My initial is MTS, by the way.
Yes, so you're very close. You could be one of them.
So I like that slide. The question I have is, so at the end of the day, I believe you are also, I believe, engineer. You're thinking about translating your research into real, practice or real solution. Um, I I hear earlier, solid tumor is very difficult to, conquer, and then your research combined with some other research, has, I see the huge potential, but I still believe there are huge, barriers, I mean, to conquer because we spend a lot of time, monies, but still, cancer is there. What would be remaining challenges you should
Mm,
overcome, and then how you could use your technology to overcome that
mhm.
quickly.
Right. So the American Cancer Society keeps these, makes an annual report every year looking at tumor incidence, so new cases, deaths, and they do it by tumor type, by state, by gender, and they keep lots of great data. And so from that data, we know that around 20% of the tumors are blood tumors, and around 80% of the tumors incident are solid tumors.
Mhm.
And you contrast that again with what Dr. Reddy mentioned about there being most of these CAR therapies being designed for blood tumors. So there really is a need to address solid tumors.
Mhm.
One of the reasons why they are hard to address is because T cells are the lack of proliferation or permissiveness in these solid tumors. So one thing is that we don't know whether they migrate into the tumor or they don't persist, which is they migrate but then die. So it can be hard to actually get T cell infiltration. The T cell infiltration that is there, we also see a heavily immunosuppressive phenotype. So people often refer to tumors as hot or cold. A
Mhm.
hot tumor is an immune responsive tumor where you have some more pro-inflammatory activation that allows you to activate the kind of immune response you need to engage in the Th1 pro-inflammatory response, where you have active CD8 T cells that can kill cancer cells, promote M1 activation of macrophages that can participate in phagocytosis, and cytokine secretion that kind of keeps that whole engine running. When tumors are cold, that means that we don't have the right type of immune cells that can cause that kind of activation. and actually contribute to immunosuppression.
Mm.
Which means suppression means no activity. So you're not getting any of that response, and you get more tumor growth. So that was just the background. So monocytes and macrophages are useful because in comparison to T cells, where they may not be migrating or proliferating as much, and tumors, and again, there's an asterisk here because it really matters by type, and there's a lot of granularity to that. But monocytes can make up large portions of the environment. And in fact, some of the resistance that we see are also because of these macrophage phenotypes that are immunosuppressive. So the studies that I'm trying to do as a cellular theranostic is how do we combine diagnostics with therapy? So I showed you a lot of the diagnostic component where we can actually use these macrophages to build sensors that we can tell, are you immunosuppressive, and how immunosuppressive are you becoming? How does a tumor environment damp those responses? And then you might be able to look at how that changes over time,
Mhm.
which is also useful because cancer is not a one-time point. It's a years-long battle.
Mhm.
But you can also then think of how you engineer those cells to then say, what if when I have activation 1, NF-kappa B activation as an example, we then say instead of expressing luciferase, you express a therapeutic drug or a cytokine, or you have some other reaction that again changes the environment. So we're trying to think about ways that we can make the environment less immunosuppressive so that we can actually have more infiltration of the right kinds of immune cells. And I think that that is a frontier that many people in cancer are trying to investigate. And one of the reasons why people look at myeloid or these monocytes and macrophages or natural killer cells as well in comparison to T cell therapies. That was a longer answer, but
Yeah. You are, I also clearly see you are very good communicator because this cancer research still, I'm researcher, but still very the complex to me. But you kind of explain very clear way. So thank you so much. I now understand much better. Let me see whether we have the question. See. Okay, I do not see the question from audience. And also I see the 11:12 Central Time. Probably we need to close.
yeah, thanks.
Okay, so let me close and then we potentially chat with the audience. Okay, so thank you all for joining and staying today. We'll meet again on December 12th, Thursday, the same time, the same Zoom link. The last webinar this year, actually from Hawaii, on the last day of the conference. We'll have the Professor Mary Dunlop at Boston University and Professor Jason Yang at Locust, New Jersey Medical School. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thank you, and I stop recording.
Thank you.